Hey, I'm Jennifer Mandel, and I'm going to talk to you today about the phylogeny of Compositae. So just give me a second to share my screen. Awesome. Okay. So again, I'm going to be speaking up about the phylogeny of Compositae, and I am Jennifer Mandel. But I just wanted to put a note here to, say that there have been so many people who have helped with this research and made it possible over the years. And so I just wanted to give a shout out to anyone and everyone who's ever loved and studied a comp. I also wanted to give a shout out to Gustavo and Mauricio and the TICA, the International Compositae Alliance, for inviting me to give this talk. I really appreciate it. And I can't give enough or loud enough shout out to Dr. Vicky Funk, who is the person who introduced me into studying really, going beyond one species, which is what I studied for my PhD, and one species of Helianthus for my postdoc. And she said to me, I can't believe, there are 25 to 35,000 species and you're just studying one. So I really want to say how grateful I am to Dr. Funk for teaching me so much about Compositae. All right, so where did we get started? So I met Vicky in 2011, and it was shortly after this, or shortly before this, that she had published the book along with many colleagues, The Systematics, Evolution, and Biogeography of Compositae. And in that, they generated a Compositae base tree using a meta-tree approach with 900 genera. It is shortened here to show just the tribes. But there were many unresolved areas of this phylogeny, and it was based on 10 plastid or chloroplast DNA loci. And here circled are, and in the dashes, are the areas where there was really a lack of resolution, a lack of understanding of what these relationships were, even on the base tree. So you can see here, sort of at the base, number one, the base of much of the majority of the species in Asteraceae, another in the Wunderlichia, etc. group, also within the subfamily Chicoriidae, and then so on up to the Helianthae, for example. And so what was really clear from this work and others was that plastid markers was not informative enough. And even ITS and ETS were informative, but there were a lot of uncertainties about paralogy. And Mike Barker's work just prior to the book being published showed that there was definitely evidence of ancient polyploidy in the family, like so many other plant families, and so like plants in general. And so this, plastid not being informative enough, and then coupled with the probably rapid radiation of the family and polyploidy, really made it challenging to resolve relationships, evolutionary relationships in Asteraceae. And then, as I said, it was about 2010 when I met Vicky in Davis, California. She was attending a Compositae genome project meeting, and I was a postdoc working on, as I said, Helianthus annuus cultivated sunflower. And Vicky had heard about next-gen sequencing, and she knew and wanted to talk to some genomics folks who were in that realm. She knew that it could hold up or had a potential for resolving some of the relationships in the family. So I'm not sure if she said this, but for sure she meant, let's give this next gen thing a go. So we did. So for the next few years, we worked on developing a bait set or a target enrichment probe set that was targeting 1,000 nuclear loci. We developed them from ESTs, which is sort of like the original RNA-seq express sequence tags. And we used about 1,300 loci that were single copy loci from Arabidopsis. And we found the best match or homologue in sunflower, lettuce, and safflower. And we designed probes, which are short 120 base RNA baits that could pull down these sequences that we wanted, or could enrich your sample for the sequences that you wanted. And so we ended up developing about 1,000 loci, 1,061 loci, and there are about 10,000 probes. And we designed this with the company that was originally called Microarray and now is Arbor Biosciences, but may have even yet a new name. But they have these MyBaits expert panels. And so our Compositae 1061 panel is available there to purchase at a reduced price, so that it's much more accessible than developing a de novo probe set. So we had these probes and we wanted to test them out in a group of species where we were pretty sure we knew the relationships. And so we did that, this shown here on the left, with about 15 taxa, even going out to Cassini, the Nastanthus species, to show that these probes could pull down or enrich loci across the family, and that the resolution for the trees was very high. And so you can see we even tested this in the genus Helianthus and then across the family. And there were, all of these nodes have stars except for one, and that's where the centropalis, and the centropalis is sister to the rest of the family. And so, we thought, well, that's interesting, but low support, so we need some more taxa there to really resolve that. That's the Chicorioideae subfamily. And so we kept adding to our trees, all the while, as I always tell people, applying for funding and getting it sometimes in small little pots of money, usually Vicki being able to write great proposals and get those small pots of funding. Always going to NSF, never getting anything, but we tried. And so as we were doing this, we started having DNAs from colleagues, collaborators, and we started adding to our tree to make it bigger and bigger. And one thing that was sort of cool and interesting to us was that subfamily Chicorioideae remained non-monophyletic, as it still does now, even when we added a lot more taxa to that. So on the right side, I'm showing a bigger sort of overview of the family. About, I think there are about 70 or so taxa in this tree. Okay, and as I said, we just kept adding samples until we covered nearly all the tribes of the family. So in a paper we published in 2019 in Proceedings of the National Academy of Sciences, or PNAS, we had 13 subfamilies, 45 tribes. We were missing 2 tribes, Polymniae and Fetae, 207 genera. And we found, a lot of new relationships. We found a lot of things we knew about, and then we found a lot of novel evolutionary relationships. For example, that, as I mentioned, that Chicorioideae remained non-monophyletic. The Cardioideae also remained non-monophyletic. And I just want to point out that this was a really great group of people to work with. Ram Thapa, who is now a postdoc in our lab, Dr. Linda Watson, who is a professor at Oklahoma State, Dr. Carol Siniskalki is a postdoc at Mississippi State, and Dr. Rebecca Deco is the chief data science officer at the Smithsonian. And I, wanted to let you know that we, of course, submitted a cover photo for the journal PNAS. Quite proud of this really cool Snesiani that Vicki's thumb is holding, very tiny there. But I am sad to inform you that this was not chosen for the cover. This was chosen for the cover. But I will note that we made a one-liner on the bottom of the cover, the evolution of sunflowers down there. But they really missed out on choosing that cover. All right, so I'll just give you a few details into what that big tree, some of the surprising relationships. So one of them was that the North American monotypic subfamily, Hecastocloideae, or Hecastocles, diverged before the Asian tribe Purdey. And that was a surprising result, although it was hinted at in some of Pinero and Funk's work, shown here, where they said it could have come before, but with the plastid loci, it showed that it diverged after Cardui. And so that's kind of interesting for the biogeographical story, which I can tell you about shortly. Something else that I already mentioned was that there was a non-monophyletic cardioideae. And so the Oldenburgii, Tarkonanthii, Dicomii, sometimes called the African Muticii, those diverged and were sister to basically the rest of Cardui. So a non-monophyletic cardioideae there. Also, the Chicorioideae, as I mentioned, remained non-monophyletic even after adding lots of taxa to the tribes in that group. So we covered all the tribes in the Chicorioideae, and we found that Chicorii is sister to Corymbium and the rest of the Asteroideae. And so that has some bearing on the classification of the family. We also wanted to use this base tree, this base phylogeny, to look again at the origins and the timing of diversifications in the family. And so for a long time, there was sort of an age for the family floating around of 40, 45 million years old. That's sort of the age that I knew about when I started studying Asteraceae. And then there was a paper that came out in 2015 by Beretta and colleagues that used a 72 million year old fossilized pollen that is attributed to the Barnardesioideae to redate using a molecular clock method to date the tree, and demonstrated that the family was probably about twice as old as we thought it was. So the family probably originated in the Cretaceous. The fossilized pollen was somewhat controversial, so there were a couple follow-ups to that. And so we wanted to use the pollen and not use the pollen in our molecular clock estimates. We also have this amazing macrofossil. It's dated to about 45 million years. It's at the split between the Barnardesioideae and the Muticioideae is where this fossil is placed. And so we used our new phylogeny to look at the origins and the timing of diversifications. And we also found a late Cretaceous origin for the Asteraceae. And then we can sort of look across the rest of the tree, and the timings and the biogeography is what I'm showing you here. And so I just wanted to point out the following animations are courtesy of / I stole them from Carol. And so that's her down there in the bottom again, showing one of our trees that we printed off on several pieces of paper. And so I'll sort of walk you through the story as it is for Asteraceae. So there's an origin in the late Cretaceous, maybe around 70 to 80 million years ago. And the origin is probably southern South America. And after that, there were movements out of southern South America into the rest of South America. And there really was not a lot of movement, or there's probably been a lot of extinction between then. The next big movement for the family is the Eocene, about 52 million years ago. And it corresponds with this EECO, or the early Eocene climate optimum. In the lower right there, I have the global temperature plotted across from the Eocene to present day. And you can see that there were several sort of spikes or climate optimums that we call them over time, one in the early Eocene, one in the mid-Eocene. And it was around this 52 million years that we have this expansion out of South America. And the route is, still maybe somewhat up in the air. If you look at the phylogeny and our biogeographical analyses, and you just look at the most likely scenario, it looks like there was probably a migration out of South America to North America to Asia, and then to Africa. But the likelihoods there, which is that pie chart that's right below the 8, while it's a higher likelihood that it's North America, there's also a good chunk of that pie chart that would be a direct route to Asia or a direct route to Africa. So, I would say the jury is still out there. But again, the most likely scenario going North America, Asia to Africa. Okay, but then what happened? So in the Eocene, from around 50 to 40 million years ago, we've a really massive amount of diversification coming out of Africa. And, I think it was Linda who coined it explosive radiations. And I think that's a good term for it, because we see essentially around 42 million years, which nicely corresponds to the middle Eocene climate optimum. We have just radiation and migrations from Africa to the rest of the globe, to Australia, to Europe, to Asia, etc., and back to South America, actually. And so this explosive sort of radiations or divergences that occurred at this time. Very cool. Okay, so all of those things that we studied or discovered in this phylogeny led us to rethink some of the classification for Compositae. I mentioned already some of the large subfamilies that were described or defined based on plastid loci appear now with a lot of nuclear genes to be non-monophyletic. And that is, for example, the Cardioideae, the Chicoroideae. So now there is potentially a Vernonioideae subfamily. And so on the right side, we've got this beautiful image that was created by Mauricio that's based on the PNAS phylogeny. And it's its own kind of phylogeny. Here you can see the basal grade down there starts with Bernardesioideae and the other South American groups. And then you have the central grade, and then the Helianthine Alliance at the top. Size of the circles is indicative of the size of that tribe. And then you can also see the number of species in the tribe in the circles. Okay, so just a few other things that we've been able to do with these data. Dr. Katie Jones was a visiting postdoc in our lab in 2016, and she did a study where she basically took anyone who had used the comp probes for whatever group, and she did a really nice assessment of the probe set and its ability to resolve relationships at multiple sort of timescales across, genera to tribes, for example, and just found some cool things from that. Genome size, negatively correlated with number of reads. One cool thing that she showed empirically was that we don't get a lot of the plastid loci when we are doing the enrichment, because we have no targets for the plastid loci, but the plastid genome is still pretty helpful for phylogenies, although that may be debated. We can debate that elsewhere. And we've sort of over the years discovered that we can spike in or add back some of the unenriched library to the capture just before sequencing, and then you can get some extra plastid loci recovered. And so Katie showed that where people who spiked or did not spike their libraries, and you recovered more targets. So that's pretty cool to show that. Also wanted to tell you about how we teamed up with some people at Kew and assessed our probe set along with the Angiosperm 353 probe set, which is a universal for flowering plants. And it, again, targets the 353 loci, and it's also available at the MyBates. So through the PAFTAL, Plant and Fungal Trees of Life project, our colleagues had sequenced a whole bunch of the same taxa with the angiosperm 353 as we had with the Comp 1061 probe set. And so Carol Siniscalchi led a project to sort of assess the lineage specific versus the universal probe set. And that just recently came out in a special issue of apps that you can check out. And so just a few things, essentially she did a whole bunch of analyses to look at the Comp 1061 captured with assemblies or sequences of the 1061, and then sort of did the opposite. So looked at things that were captured with species that were captured with the 1061, but assembled them with the angiosperm 353 and vice versa. And a number of trees I could show you, but I kind of wanted to just give the bottom line there is that the angiosperm 353 had an average of 287 loci with very low levels of paralogy, whereas the Compositae had an average of 721 loci, but with a lot more 9 to 46% potential paralogs. And so we also discovered that there are 30 shared loci in the probe set. So that allows you to be able to combine the data. And a lot of people are testing out this idea or doing in the lab capturing with the angiosperm and a lineage specific probe set. So essentially doing two types of captures in one reaction and sequencing that. And so I think that's going to be super promising and allowing people to sort of, as the paper that we're not co-author, but there's a special issue paper called The Best of Both Worlds. And so combining the universal with the lineage specific, I think is going to be a really promising thing for the future. And so again, I just wanted to sort of, show there's a lot of people who are working with the Asteraceae and the probe set. Here's just some of their faces. Of course, I always want to tell you, you can be next. You can study a genus or a tribe or a whole subfamily if you want. And what we're working on next is integrating data. So we're really interested in integrating different probe sets and adding other things like morphology and whole genome duplications and paleopolyploidy. Really interested in getting at the genomic basis of these key innovations. So I've always been told, always been read, the capitulum, the pappus, the secondary chemistry. These are things for Asteraceae that likely really led to its success. And we are now in the moment where we can use whole genomes to be able to maybe get at the functional genes that are underlying these traits. It's really an amazing time to be studying Asteraceae. And along with that, we have various genome projects that are running using long read technologies. Centropalus is one of them that I have pictured here. And then now comes the plugs. I want to plug our TICA talks, which are the last Mondays of the month usually, and you can find out about them at Twitter at TICA talks. There's also a link on the website, the compositae.org, that has the information. Here's just one of them. Of course, we're all on Zoom and happy with many sunflowers in the background. So it's been a really awesome, it started in January. It's been a really awesome opportunity to hear about people all over the world. And, some people even wake up at 4 a.m. to give us talks. So that's been pretty awesome. And then finally, a plug for the TICA newsletter, which is the Capitulum. And our first issue should be out very soon, if not already, by the time we're listening to this presentation. And I'll point out the editorial board, Mauricio Bonificino, Benoit Lual, hope I didn't mess that up. I think I probably did. Me, Abigail Moore, Erica Moore, who is a graduate student in our lab, and Lizzie Roble, sorry. And submissions can be made with going to compositae.org/capitulum, and you can find out all about the newsletter and think about contributing and reading the newsletter. So with that, again, I want to thank so many people including all those people who ever loved and studied a composite, as well as all these folks here and funding sources for us studying this. And now I will live take your questions.